History of coronavirus. The history of coronaviruses is a reflection of the discovery of the diseases caused by coronaviruses and identification of the viruses. It starts with the first report of a new type of upper respiratory tract disease among chickens in North Dakota, U.S. in 1931. The causative agent was identified as a virus in 1933. By 1936, the disease and the virus were recognized as unique from other viral disease. They became known as infectious bronchitis virus IBV, but later officially renamed as avian coronavirus. A new brain disease of mice, murine encephalomyelitis, was discovered in 1947 at Harvard Medical School in Boston. The virus causing the disease was called JHM after Harvard pathologist John Howard Mueller. 3 years later, a new mouse hepatitis was reported from the National Institute for Medical Research in London. The causative virus was identified as mouse hepatitis virus MHV. In 1961, a virus was obtained from a schoolboy in Epsom, England, who was suffering from common cold. The sample designated B814 was confirmed as novel virus in 1965. New common cold viruses assigned 229 collected from medical students at the University of Chicago were also reported in 1966. Structural analyses of IBV, MHV, B814, and 229 using transmission electron microscopy revealed that they all belong to the same group of viruses. Making a crucial comparison in 1967, John Almeida and David Tyrrell invented the collective name coronavirus, as all those viruses were characterized by solar corona-like projections called spikes on their surfaces. Other coronaviruses have been discovered from pigs, dogs, cats, rodents, cows, horses, camels, beluga whales, birds, and bats. As of 2020, 39 species are described. Bats are found to be the richest source of different species of coronaviruses. All coronaviruses originated from a common ancestor about 293 million years ago. Zoonotic species such as severe acute respiratory syndrome-related coronavirus SARS-CoV, Middle East respiratory syndrome-related coronavirus MERS-CoV, and severe acute respiratory syndrome-related coronavirus 2 SARS-CoV-2, the etiological agent of the COVID-19 pandemic. Discovery of chicken coronavirus. Arthur Frederick Schock and Merle C. Fawn at the North Dakota Agricultural College were the first to report what was later identified as coronavirus disease in chickens. Their publication in the Journal of the American Veterinary Medical Association in 1931 indicates that there was a new respiratory disease that mostly affected 2-day-old to 3-week-old chickens. They referred to the disease as an apparently new respiratory disease of baby chicks. The symptoms included severe shortness of breath and physical weakness. The infection was contagious and virulent. It was easily transmitted through direct contact between chickens or experimental transfer of the bronchial exudates from infected. Maximum mortality recorded was 90%. The causative pathogen was not known. Charles D. Hudson and Fred Robert Bowdett at the New Jersey Agricultural Experiment Station in New Brunswick, New Jersey, put forth a hypothesis in 1932 that virus could be the cause and introduced the name as virus of the infectious bronchitis. But this was a misattribution because at the time another related disease, known as infectious laryngotracheitis, was reported that exhibited almost similar symptoms but mostly affected adult chickens. As Bowdett later recalled in 1937, the disease he described was infectious laryngotracheitis, saying infectious laryngotracheitis is said to be the correct name for this disease rather than infectious bronchitis. Moreover, the gasping symptom ordinarily accepted as typical of the disease is also a prominent symptom in infectious bronchitis. The names infectious bronchitis and infectious laryngotracheitis were till then used synonymously and interchangeably. Unaware of the developments, Leland David Bushnell and Carl Alfred Brandley at the Kansas Agricultural Experiment Station studied a similar case, which they called gasping disease due to the apparent symptom. They had known the disease since 1928. Their report in 1933 titled Laryngotrichates in Chicks published in the Poultry Science indicated a clear distinction of infectious bronchitis from infectious laryngotrichates, as the main organ affected was the bronchi infection resulted in severe gasping and swift death due to inability to eat food. It was also found that the pathogens could not be bacteria or protozoans, as they passed through membrane's Berkefield filter that would block those pathogens. The isolation and identification of the pathogen as a virus was reported as in several experiments. We have reproduced the disease in chicks by the intratracheal, subcutaneous, and intraperitoneal injection of Berkefeld filtered material. The chicks developed typical gasping symptoms after various periods of incubation, different groups of chicks first showing symptoms in 6, 17, 19, etc. days after receiving the filtrate. The disease may also be transferred by means of filtrates of spleen, liver, and kidney tissues, and by the transfer of bacteriologically sterile blood. This was the discovery of infectious bronchitis virus IBV. But Bushnell and Brandy made an erroneous remark by saying the symptoms and lesions in the chicks caused by IBV are similar to those seen in so-called laryngotrichates of adult birds and are probably due to the same agent. In 1936, Jerry Raymond Beach and Oscar William Shalm at the University of California, Berkeley, each had discovered infectious laryngotrichates virus in 1931. They concluded that it was found that chickens that recovered from an infection with one of the two strains of virus were refractory to further infection with either strain. It was also found that the sera from chickens that have recovered from an infection with one strain of the virus would neutralize virus of either strain. These results show the identity of the two strains of virus. Chickens refractory to infection with this virus were shown to be susceptible to the virus of laryngotrichates. Likewise, chickens refractory to the latter virus were susceptible to the former. These results demonstrate that the two viruses are distinct from one another. Hudson and Bowdit later in 1937 were able to culture IBV for the first time using chicken embryos. This specimen, known as the Bowdit strain, became the first coronavirus to have its genome completely sequenced in 1987. Discovery of mouse coronaviruses. Francis Sargent Cheevers, Joan B. Daniels, Olwyn M. Pappenheimer, and Orville T. Bailey investigated the case of brain disease murine encephalitis at the Department of Bacteriology and Immunology of Harvard Medical School in Boston in 1949. Two laboratory mice Schwenker strains of 17 and 18 days old had flaccid paralysis and died. By then it was known that murine encephalitis was caused by a picoinavirus, called Thaler's virus, which was discovered by Max Thaler at the Rockefeller Foundation in New York in 1937. But the Harvard scientists found that the two mice had unusual symptoms other than brain damage demyelination. The mice had no visible illness or diarrhea, which usually are associated with murine encephalitis. In addition, the causative virus was isolated from different organs including liver, spleen, lungs, and kidneys. This indicated that brain was not the primary target organ. Liver was particularly affected with severe necrosis, indicating hepatitis. The new virus was named JHN, after the initials of John Howard Mueller. In the autumn of 1950, Alan Watson Gledhill and Christopher Howard Andrews isolated the causative virus, which experimentally was highly infectious to healthy mice. They named the virus as mouse hepatitis virus MHV. Gledhill called the experiments on the highly infectious nature of the virus as a bizarre discovery. John A. Maurice at the National Institutes of Health, Bethesda, discovered a new mouse virus, named H747 in 1959 from samples in Japan. When he compared the virus with JHM and MHV using serological tests, he found that they were both antigenically related. Discovery of human coronaviruses. Human coronaviruses were discovered as one of the many causative viruses of common cold. Research on the study of common cold originated when the British Medical Research Council and the Ministry of Health established the Common Cold Research Unit (CCRU) at Salisbury in 1946. Directed by Andrews, the research laboratory discovered several viruses such as influenza viruses, parainfluenza viruses, and rhinoviruses that cause common cold. David Arthur John Tyrrell joined CCRU in 1957 and succeeded Andrews in 1962. He developed a technique for growing rhinoviruses using nasal epithelial cells for the first time in 1960. His team soon after developed a concept of broad categorization of common cold viruses into two groups. One group, called H strain, could be maintained only in human embryo kidney cell culture, and another group, designated M strain, could be maintained both in human embryo kidney cell culture and monkey embryo kidney cell culture. By then, many common cold viruses could be grown in either of these cell cultures and were accordingly classified as M or H strain. During 1960-1961, Tyrrell's team collected throat swabs from 170 schoolboys having common cold at a boarding school in Epsom, Surrey, England. Among few samples that could not be cultured in any of the culture media, a specimen designated BATE-114, collected on 17 February 1961, was particularly infectious among healthy volunteers. There was no evidence whether the pathogen in BATE-114 was a bacterium or a virus, as all bacterial and viral culture methods available showed negative results. It could only be maintained in human tracheal culture and experimentally passed on to healthy volunteers by nasal inoculation. In 1965, they were able to confirm that the pathogen was a filter-passing virus susceptible to ether treatment, indicating a lipid envelope of the virus able to induce cold in antibiotic-treated volunteers, indicating it was not a bacterium, and cultured in human embryo tracheal epithelial cell culture. Serological tests, antigen-antibody reactions further indicated that the virus was not reactive to antibodies serotypes of any known viruses at the time. Reporting in the British Medical Journal, Tyrrell and Malcolm L. Bino wrote their conclusion as, after considerable initial doubts, we now believe that the BATE-114 strain is a virus virtually unrelated to any other known virus of the human respiratory tract, although, since it is ether labile, it may be a myxovirus. But they contradicted themselves regarding the identity of the virus, as they mentioned in the experimental results, saying, "It was concluded that BATE-114 did not belong to any of the serotypes of myxovirus used, but might be distantly related to influenza C or Sendai viruses." In an independent research in U.S., Dorothy Hammer and John J. Prochnow studied respiratory tract infection among medical students at the University of Chicago. In 1962, they obtained 5 samples that were associated with very different symptoms, causing mild cold only, and could be cultured only in secondary human kidney tissue, in contrast to other cold viruses which could be maintained in monkey embryo kidney cell culture. Serological tests indicated they were not myxoviruses or Orthomyxoviridae. They presented their discovery as a new virus isolated from the human respiratory tract in the Proceedings of the Society for Experimental Biology and Medicine in 1966. They further studied one sample, designated 229E, grown in human diploid cell culture women as 38, and described its developmental stages using transmission electron microscopy to show that it was new type of virus. Discovery of the structure. Viruses cannot be seen normally with light microscopes. It was only with the development of electron microscopy that viruses could be visualized and structurally elucidated. Reginald L. Reagan, Jean E. Hausser, Mary G. Lilly, and Arthur H. Craig Jr. of the University of Maryland were the first to describe the structure of coronavirus using the transmission electron microscopy. In 1948, they reported in the Cornell Veterinarian that IBV was spherical in shape, and some of them had filamentous projections. But the images were difficult to interpret due to poor resolution and low magnification at x 28,000. Their subsequent studies did not show any striking properties from other viruses. An important advancement was made by Charles Henry Domermuth and O. F. Edwards at the University of Kentucky in 1957 when they observed IBV as ring or donut-shaped structures. D. M. Berry at the Glaxo Laboratories, Middlesex, UK, with J. G. Cruikshank, H. P. Chew, and R. J. H. Wells at the University of Cambridge published more comprehensive and better electron microscopic images in 1964. 4 strains of IBV, including Baudet strain, were compared with influenza virus, with which they share most resemblance. In contrast to influenza virus, in which the projections were small and straight, all IBV strains had pear-shaped projections, which were named the spikes and described as these spikes were often seen over part of the surface only and were less densely packed than those seen in influenza viruses. They varied considerably in shape. Commonly, they appeared to be attached to the virus by a very narrow neck and to thicken towards their distal ends. J. F. David Ferreira and R. A. Manaker from the National Cancer Institute, Bethesda, were the first to study the structure of MHV in 1965. They also observed the surface projections as on IBV, stating the outer surface of the particle is covered by spicules. In 1966, Tyrrell sought the help of Anthony Peter Watterson at the St. Thomas's Hospital Medical School in London, who had recruited June Dalziel Almeida as an electron microscopist. Almeida had studied IBV and MHV, finding them as structurally distinct viruses, but her manuscript was rejected upon a referee's decision that the microscopic images were of influenza virus and lacked novelty. Tyrrell supplied the human virus samples from which it was possible to ascertain the viral structure. Almeida and Tyrrell published their findings in the April 1967 issue of the Journal of General Virology, in which they concluded, probably the most interesting finding from these experiments was that 2 human respiratory viruses, 229E and 814, are morphologically identical with avian infectious bronchitis. Their biological properties, as far as they are known, are consistent with this. Both the human viruses are ether-sensitive, as is avian infectious bronchitis 229E, have a similar size by filtration, and multiply in the presence of an inhibitor of DNA synthesis. In 1967, Kenneth McIntosh and co-workers at the National Institute of Health, Bethesda, reported the structure of common cold viruses they collected from fellow workers during 1965–1966. They found 6 of their samples had common characters with 814. 2 samples designated OC38 and OC43 as the number of specimen in organ culture were particularly virulent and caused encephalitis in experimental mice. They compared the structure of one of their samples numbered 501 OC43 with those of 229E IBV and influenza virus. It was so identical to IBV that they called the human viruses as IBV-like viruses. They made a definitive description as all IBV-like viruses, 229E and IBV itself, show the following characteristics. 1. An overall diameter of 160 mm with a variation of plus 440 mm. 2. A moderate pleomorphism with resultant elliptical, round, or teardrop shapes but no filamentous. Invention of the name and history of the taxonomy. By mid-1967, it was recognized that IBV, MHV, 814 and 229E were structurally and biologically similar, so that they form a distinct group. Tyrrell met Watterson and Almeida in London to decide on the name of the viruses. Almeida had earlier suggested the term influenza-like because of their resemblance, but Tyrrell thought it inappropriate. Almeida came up with a novel name, coronavirus. Tyrrell wrote of his recollection in Cold Wars, the fight against the common cold in 2002, even though we could only base our judgment on the electron microscope images, we were quite certain that we had identified a previously unrecognized group of viruses. So what should we call them? Influenza-like seem a bit feeble, somewhat vague, and probably misleading. We looked more closely at the appearance of the new viruses and noticed that they had a kind of halo surrounding them. Recourse to a dictionary produced the Latin equivalent, corona, and so the name coronavirus was born. Proposal of the new name was submitted to and accepted by the International Committee for the Nomenclature of Viruses (ICNV), established in 1966. 16 November 1968 issue of Nature reported the justification by Almeida, Berry, C. H. Cunningham, Hammer, M. S. Hofstad, L. Malaxi, McIntosh, and Tyrrell as particles of IBV are more or less rounded in profile, although there is a certain amount of polymorphism. There is also a characteristic fringe of projections 200 along. This appearance, recalling the solar corona, is shared by mouse hepatitis virus and several viruses recently recovered from man, namely strain B814, 229E, and several others. In the opinion of the 8 virologists, these viruses are members of a previously unrecognized group, which they suggest should be called the coronaviruses, to recall the characteristic appearance by which these viruses are identified in the electron microscope. Coronavirus was accepted as a genus name by ICNV in its first report in 1971. IBV was then officially designated the type species as Avian, infectious bronchitis virus, but renamed to Avian coronavirus in 2009. Mouse hepatitis virus, approved in 1971, was merged with rat coronavirus discovered in 1970 as murine coronavirus in 2009. 229E and OC43 were collectively named human respiratory virus but merged as human coronavirus 229E HCOV-229E in 2000. The first discovered human coronavirus, B814, was antigenically different from 229E and OC43, but it could not be propagated in culture and was exhausted during experiments in 1968, thus was excluded in taxonomy. Coronaviridae was adopted as the family name in the ICNV soon after renamed International Committee on Taxonomy of Viruses, ICNV second report in 1975. 229E and OC43 were together named human respiratory virus in the ICNV first report. The species was split into human coronavirus 229E HCOV OC229E and human coronavirus OC43 HCOV OC43 in 1995. While HCOV OC229E is retained as a valid species, HCOV OC43 was merged with porcine hemagglutinating encephalomyelitis virus discovered in 1962, bovine coronavirus discovered in 1973, D3 human enteric coronavirus discovered in 1970. Owing to increasing number and diversity of new species discovered, CTV split the genus coronavirus in 2009 into 4 genera: alpha coronavirus, beta coronavirus, delta coronavirus, and gamma coronavirus. As of 2020, there are 39 species of coronaviruses under the family Coronaviridae. There are 7 human coronaviruses, while 32 species are those of pigs, dogs, cats, rodents, cows, horses, camels, beluga whales, birds, and bats. Other human coronaviruses. Human coronavirus and L63 HCOV and L63. HCOV and L63 was discovered in January 2003 from a 7-month-old baby in Amsterdam, the Netherlands. The baby was suffering from bronchiolitis, coryza, conjunctivitis, and fever. A year later, a comprehensive analysis of nasal swab samples was done, from where it was found that a sample from an 8-month-old boy diagnosed in 1988 with pneumonia had a similar virus HCOV and L. The virus was independently described in 2005 as HCOV and H, following a discovery among a group of children having respiratory infection in New Haven, Connecticut, U.S. The origin of the virus remains a mystery, but it is closely related to tricolor bat perimyotes subflavus coronavirus and can survive in bat cell lines, suggesting that it is derived from animals zoonotic. Human coronavirus HKU1 HCOV HKU1 KU1 HCOV HKU1 was discovered from a 71-year-old man in Hong Kong, China, who was suffering from pneumonia in January 2004. When samples nasopharyngeal aspirates from pneumonia patients collected between April 2004 to March 2005 were analyzed in 2006, it was found that 13 individuals had HCOV HKU1. The same year, the virus was subsequently reported from Australia, Europe, and US. Zoonotic coronaviruses. Coronaviruses that are transmitted from animal zoonoses are clinically the most important human coronaviruses, as they are responsible for a series of global epidemics. There are two species of such coronaviruses. 1. Severe acute respiratory syndrome-related coronavirus. Two distinct viruses are known under this species, namely SARS-CoV and SARS-CoV-2. SARS-CoV emerged as an acute respiratory syndrome in Guangdong Province, southern China, during 16 November 2002 to 28 February 2003. The syndrome was accompanied by pneumonia that was fatal in many cases. The infection was believed to have been contained in China, but an infected individual carried it to Hong Kong on 21 February and spread it in the hospital. The first clinical case outside China was reported on 26 February 2003 in Hanoi, Vietnam. It rapidly spread to Southeast Asia, North America, and Europe. The World Health Organization, who notified an epidemic alert on 6 March 2003, referring to the disease as severe acute respiratory syndrome. The virus was identified as a novel coronavirus from Hong Kong in April, from Toronto in May, and at the Centers for Disease Control and Prevention (CDC) in US in May. In October, the samples from Guangdong were established as the prototype specimens, and the name SARS coronavirus SARS-CoV was introduced. ICTV approved it as severe acute respiratory syndrome coronavirus in 2004 and renamed it severe acute respiratory syndrome-related coronavirus in 2009. By mid-July 2003, the infection subsided, and by then it had spread to 28 countries, infecting 8,096 people and causing 774 deaths. In October, it was found that the infection was acquired from the masked palm, Civets paguma larvata, from a live animal market in Guangdong. Further studies in 2005 showed that civets were the intermediate reservoirs of the virus, and horseshoe bats Rhinolophus species were the natural hosts. Infection with SARS-CoV-2 was known from cases of atypical pneumonia in Wuhan, China. The Wuhan Municipal Health Commission reported 27 individuals having viral pneumonia on 31 December 2019. The first known case was recorded on 12 December. The first case outside China was in Thailand on 13 January. Who adopted the name of the disease as coronavirus disease 2019 COVID-19 on 11 February 2020 and used 2019 novel coronavirus or 2019 nCoV for the virus. On 2 March 2020, ICTV published the formal description and gave the official name as severe acute respiratory syndrome-related coronavirus 2 SARS-CoV-2, who declared the infection as pandemic on 11 March, and since then has spread around the world, affecting over 521 million people and resulting in more than 6.26 million deaths. The origin of the virus is not known. Malayan pangolins Manis javanica, which are available in the live animal market in Wuhan City, has been studied as a probable source, as the virus is closely related to the pangolin coronavirus. Genetic evidence that it bears 93% nucleotide similarity with a novel coronavirus of Malayan horseshoe that Rhinolophus malayanus, a 96% identity with bat SARS-like coronavirus RA-TG13 of intermediate horseshoe bat R. Affinis indicates that it probably originated in bats. 2. Middle East respiratory syndrome-related coronavirus. In April 2012, the Ministry of Health, Jordan, reported an outbreak of acute respiratory illness affecting 11 people at a hospital in Zarqa. On 13 June 2012, a 60-year-old man having the symptoms was admitted to Dr. Solomon Faki Hospital in Jeddah, Saudi Arabia. He was diagnosed with acute pneumonia and died on 24 June due to progressive respiratory and renal failure. His sputum sample showed the presence of coronavirus very similar to bat coronaviruses HKU4 and HKU5. The virus was named HCOV after Erasmus Medical Center in Rotterdam, the Netherlands, where it was identified. Retrospective study of samples from the Jordan hospital revealed that the diseases and the virus were similar. Who referred to the virus as the Middle East Respiratory Syndrome Coronavirus MERS-CoV on 23 May 2013. ICV announced the name as Middle East Respiratory Syndrome-related coronavirus on 15 May 2013. In 2013, a study revealed that the virus was 100% genetically identical to the coronavirus of the Egyptian tomb, that Taphosis perforatus coronavirus HKU4 from Bisha, Saudi Arabia, indicating its origin. In 2014, it was established that the virus was transmitted to humans by dromedary camels, which act as the intermediate hosts. By December 2019, the infection was confirmed in 2,499 individuals with 858 deaths, 34*3% mortality from 27 countries covering all continents. Other animal coronaviruses. Feline infectious peritonitis virus, a viral infection in pigs called transmissible gastroenteritis, which was characterized mainly by diarrhea and vomiting and associated with high mortality, was first recognized by Leo P. Doyle and L.M. Hutchings in 1946. A.W. McClurkin isolated and identified the virus in 1965. The virus was named transmissible gastroenteritis virus of swine in the ICMA first report and changed to porcine transmissible gastroenteritis virus PTGV in the second report in 1976. A new disease that caused inflammation of the abdomen, peritonitis, in cats was reported in 1966. The virus was identified in 1968 and was named by ICMA in 1991 as feline infectious peritonitis virus. In 1974, a new coronavirus was discovered from U.S. military dogs and was named by ICMA in 1991 as canine coronavirus. As the molecular and antigenic relationship of the three viruses were later established, ICMA merged the three viruses into alpha coronavirus 1 in 2009. Porcine epidemic diarrhea virus. An acute infectious diarrhea was first known in England in 1971. The infection was specifically among fattening pigs and sows. It was referred to as TO for the other one or TG etwo for transmissible gastroenteritis type 2, as the symptoms were similar to transmissible gastroenteritis. Other than causing rapid and acute diarrhea, it was not a fatal disease. The case was first reported by J. Oldham in pig farming in 1972 using the term epidemic diarrhea. A second outbreak occurred in 1976 and was called porcine epidemic diarrhea. It eventually spread throughout Europe. M.B. Penseert and P. de Boek at the Ghent University, Belgium, isolated and identified the new coronavirus in 1978 and designated it CV777. It eventually spread throughout Europe. ICMA named the virus porcine epidemic diarrhea virus in 1995. An epidemic broke out from China in 2010 that spread throughout the world. A virulent strain emerged in U.S. between 2013 and 2015. It affected pigs of all ages, and mortality was as high as 95% among the suckling piglets. Another severe outbreak occurred in Germany in 2014, that spread to other European countries. Bat coronaviruses. Reagan and his colleagues at the University of Maryland were the first to investigate bats as a potential sources of coronavirus in 1956. They experimentally inoculated 44 cave bats or little brown bats Myotis lucifugus with IBV and found that all of them developed the symptoms of infectious bronchitis. Their report reads, 50% of the bats exposed to the infectious bronchitis virus showed symptoms or death in the intracerebral, intraperitoneal, intradermal, intracardiac, and intraocular groups, 75% in the intranasal and intrarectal groups, 100% in the intraoral group, and 25% intralingual. But nothing was known of the real nature of bats as reservoirs of coronaviruses until the epidemic of severe acute respiratory syndrome of humans in 2002/2003. Since the identification of SARS-CoV was identified in early 2003 and horseshoe bats as their natural hosts in 2005, bats have been extensively studied. Among all coronavirus hosts, bats are known to harbor the most variety, with more than 30 species identified. According to Diversity Estimate, there may be 3,200 species of coronaviruses in bats. Evolutionary history. According to phylogenetic estimate, all coronaviruses evolved from the most recent common ancestor that lived around 190 to 489, with a mean of 293 million years ago. The four genera split up around 2,400 to 3,300 years ago into bat and avian coronavirus ancestors. That coronavirus gave rise to species of alpha coronavirus and beta coronavirus that infect mammals, while avian coronavirus produced those of gamma coronavirus and delta coronavirus that infect birds. Zoonotic coronaviruses emerged recently. For instance, SARS-CoV was transmitted from bats in 1998, 4.8 years prior to the outbreak, and diverged from bat coronavirus in around 1962. SARS-CoV-2 evolved from bat coronavirus in around 1948. References